now what we start with next is going to be ELISA. First you need to know that this is an ELISA plate, what you have in front of you. This is an ELISA plate under which you can see there are 12 wells in this direction and there are 8 wells in this direction. So total becomes 12 into 8 and that is going to be 96 wells. This could be a question in path, micro or biochem, wherever ELISA is used. Okay, so 12 and 8, total 96 wells, 96 testings can be done at the same time. Such a useful thing. But what is ELISA? Enzyme linked immunosorbent assay. So means it's an assay, it's a test in which I'm doing some immuno-antigen antibody stuff, but that is with the help of an enzyme. So first I want you to ratta lagao 3 things, then I'll tell you the types of ELISA. In this, if you ask me what is the enzyme is HRP. If you want to know the full form, horse radish peroxidase. It is going to be horse radish peroxidase. Peroxidase tells me that yeah, it is an enzyme. Then you have a substrate called hydrogen peroxide, H2O2. Enzyme will always break something. Na? So substrate kya hai? That is H2O2. And in the end, how will you get to know test is positive or negative? You should have some color. So the color is some form of benzidine that is used. Over here they are using tetramethyl benzidine. So 2-3 things you ratto. Which is the enzyme? Peroxidase. What is it going to break? H2O2. And what is the color? There is a color known as benzidine. So now what am I going to tell you? There are 4 types of ELISA that we must know for the exam, which I'm sure all of you know. Direct ELISA, indirect ELISA, sandwich ELISA, competitive ELISA. Now let's start one by one. Direct में क्या measure कर रहे हो? पहले तो रट लो. Direct में what are you measuring? Direct में you're measuring the antigen. Sandwich में you're measuring the antigen. Competitive में you're measuring the antigen. Means where are you testing the antibody in the indirect ELISA? Point number 1. अब डिटेल में चलते. You should know now what am I trying to look for in the patient. So in all of them, you are trying to look for antigens in the patient. The only one where you're trying to measure antibodies in the patient is indirect ELISA. Now let's go into the details. So what is a direct ELISA? Starting from the first one. So you'll say, sub mein ma'am, it is antigen detection. This means this is that ELISA well. If I want to test for antigen, this means if I want to test for the antigen, this means the first thing that I'll add is the patient sample. I want to find out antigen in his sample na, so I will add a sample first. Then patient ka kaam to khatam. He has given me his antigen. Now I will add an antibody in the lab. You know what I'll do? Smartness that I'll do. I will combine that antibody with an enzyme. Which enzyme are we dealing with? Horseradish peroxidase. So for example, let's assume patient mein antigen tha. Okay? If the patient had the antigen, if I'll add an antibody with an enzyme, will an antigen-antibody reaction occur? Yes. Antigen-antibody reaction hoga. How will I get to know? You say, ma'am, give the substrate for this enzyme, add the substrate for this enzyme, that is H2O2. This enzyme will then go and break the substrate, add some color. You will get to know what color. You add benzidine and you will get a color positive. So simple. If there is an antigen in the patient's blood and you have an antibody the antibody will get added to it, get attached to it. If you add an enzyme, the enzyme will break this H2O2. H2O2 will change the color and the color of benzidine will come out. So simple. Antigen hai, to color aayega. Means I can even measure the intensity. How much of color is coming will tell me how much of antigen is coming. That is, so you must have heard that reports always give you quantity. Antigen is 4 ml, 5 ml, 3 microliter. How? Because that's on the basis of the color. More color matlab there was more antigen that was present in the well. So simple. Now you know if you want to test an antigen, that is direct ELISA. But what is indirect ELISA? Indirect ELISA mein, what do you want to test, guys? Indirect ELISA mein, you are gonna basically measure the antibody. You are gonna measure the antibody in the patient. So now you will say that, ma'am, niche to antigen dalna padega. So now you will add antigen on your own. From your lab, you have these machines ka manufacturers, they'll give you antigen. Then you will add the antibody from the patient. You will add the antibody from the patient sample because you want to test whether does he have the antibody or not. So you'll say, antigen ma'am, I contributed on my own. Then I added the patient sample. Then to confirm this, I did the same process again. I added another antibody. This time I had conjugated that with an enzyme. That enzyme went and broke the substrate H2O2 that I added, and finally I got the color. So more the color that I get means more was the antibody in the patient's sample. So ultimately I realized adding an antibody which is linked with an enzyme, and the enzyme will break the substrate H2O2 and the color will come out. That was there in direct ELISA also. That is there in indirect. That will be there in every ELISA. What are you detecting will change. In direct ELISA, direct patient ka sample dala, khatam. That's all that you had to do because you were testing antigen. Whereas in indirect ELISA, first you add an antigen, then you add the patient sample indirectly because now you want to test for antibodies. Let's come to the third one, sandwich. Ab sandwich banao. Sandwich kaisa banega? There are two slices of bread. And there has to be butter or some thing in between for a sandwich. So you will say, ma'am, sandwich matlab two slices of bread matlab antibody idhar, antibody idhar. There is antibody on both sides and you are basically having an antigen in the center. So ye ho gaya antibody, ye ho gaya antibody, and this is going to be an antigen in the center. So tell me, for everything you were trying to measure the antigen only, na? So you will say, ma'am, patient mein to kya measure karna hai? Antigen measure karna hai. This means this antibody you will have to add, and the upar wala antibody also you will have to add on your own. Patient say you're only going to get the antigen. So you add an antibody first, you capture the patient's antigen, then you add an antibody again, and after that the rest of the story is the same. Na? You add that antibody which has an enzyme is going to break the substrate H2O2, then the color is going to come. So sandwich ELISA is also going to be a variety of direct ELISA. Why? Because it is going to do antigen detection, but antigen ko humne sandwich bana diya. Either antibody, and then the rest of the story is the same. Coming to the last one, competitive ELISA. This was also for antigen detection, but here I am gonna create a competition. Competition means think of the topper of your class. He used to come to the class and say, "Arey, kuch nahi padha, I have not studied anything. I was slept off yesterday. I had gone for a movie." But what was he doing? He was actually sitting locked the hostel room and was studying or sitting in some library. So pehle se he has done his homework and come, hai na? So what am I doing over here? If you want to test for antigen, like you say that, ma'am, I want to test for free testosterone levels in a male patient. How much of testosterone does he have? So you want to test for some kind of antigen in a person. What will you do? Whatever antigen you had, usse pehle hi bahar. Finish the course in the library. Don't bring it to the classroom. Class mein aane se pehle those students who read and come na. So what have you done? You have taken the antigen from the patient and you have already added an antibody to it. So you have already done an antigen-antibody reaction outside the well. Alag se test tube mein kar diya. Pehle se course khatam. Now just imagine if you add this antigen-antibody mixture to an ELISA plate. And I know every ELISA plate I will be coating it with antigens. I have already coated it with antigen, but you'll say, ma'am, when that person has already finished his syllabus outside the class, why will he come and learn anything new? Even if you put him in the class, he's gonna chill. He will show everyone that, I don't study. Why? Because he has already finished his syllabus outside. So if antigen-antibody reaction as a competition has occurred outside, when it will come inside this well, it will keep floating like this. It will try to show chill, I'm floating around. I'm not going to show this reaction. Now when you do that test of yours, that is, you used to add the secondary antibody and the enzyme and the color, all of that. So you will say that, ma'am, if for example there were 5 antigens in the beginning and you ended up adding 10 antibodies, assuming patient ke paas 5 antigens the and you had added 10 antibodies. So can you say 5 to consume ho gaye? How many antibodies were left extra? 5 antibodies were left extra. You ended up adding little more substrate, little more reagents. तो वो 5 antibodies में तो कुछ reaction दिखाया होगा, तो कुछ color तो आएगा. Little color will come. So now this works as an inverse. Competition में उल्टा चलेगा. If you get a lot of color, this means all your antibodies had basically gone free. Antigen था ही नहीं. So all these antibodies had the time in life to come and study. बाहर कुछ competition था ही नहीं, बाहर कोई पढ़ाई करा ही नहीं. That is why all the antibodies had come and they had gotten bound over here. That is why they gave so much of color. So if you get too much of color, this means बाहर homework हुआ ही नहीं था. If you start studying too much in the ELISA plate, if you start studying too much in the class, this means there was no homework that you did beforehand. On the other hand, if you stop getting any color, if you don't get any color, you will say, ma'am, there is no one studying inside this, no reaction happening inside this ELISA plate. This means all the reaction has occurred outside. This means the antigen was in excess. Now you are understanding the competition कि सब बाहर ही हो गया reaction. So simple, this works in an inverse relation, and the inverse relation is that if you are getting a lot of color in the end, this means the concentration of the antigen was low. And if you are not getting color in the end, this means the concentration of the antigen is high. It's simple. If you are not studying in class, this means you have done your padhai bahar. And if you are studying too much in class, this means you have not done your padhai bahar. Okay, that is what is a competition. So now, when we do a final summary, didn't I tell you direct, sandwich, competitive? In everything you study the antigen, but in everything, if you remember, more the color you get, more is the antigen. More the color you get, more is the antigen. But in competition, it is ulta. More the color you get, less is the antigen. And what were you doing in indirect? You were testing for antibody. Now the only difference that I'm going to do, this was all ELISA. There is something also known as CLIA. CLIA is known as chemiluminescence immunoassay. So firstly, what did I say? Immunoassay. Ma'am, ELISA में भी immunoassay था. But over here, you are using chemiluminescence. What does that mean? That means that you are not going to get a color in the end. You are going to get something shiny in the end. अभी तक तो you were saying color आया, color नहीं आया, but now you are saying some chemical light is gonna come. If some light is coming, this means you have not used that benzidine. In ELISA, the color that you were using, was benzidine. You were using color, that is why you were getting color. But if you start using some luminal esters, you will start getting light signals. Baki sab same, antigen, antibody, platelet, sub same. Only difference is that in ELISA you are dealing with colors, and in chemiluminescence you are dealing with something luminescence, luminal light, shine. That's the difference. Coming to the next, I'm sure all of you have also heard of immunofluorescence. What is it about immunofluorescence that you have to know? There is a direct immunofluorescence or direct fluorescent testing. Then there is an indirect fluorescent testing. So sabke bare mein we are going to study. So we will repeat it once again. Direct and indirect. Okay? Now I tell you I teach it a little different in comparison to how others have taught it, because this one slide will give you everything. Lag to same raha hai, ma'am. It's looking antigen, antibody. In the end, you have added some light wala signal because fluorescence, everything is looking the same. Like I said, direct fluorescent testing is basically for testing the antigen. So now tell me, patient mein what are you trying to find? Antigen. So this is the patient ka sample that you have added. Patient ka antigen. What will you add on top of it? You will add an antibody and you will add some fluorochrome signal. You will add some fluorescent signal. Which is the fluorescent dye which is most commonly used for this process? It is FITC. Wherever they ask you fluorescent dye in any machine that is being used in path, micro and biochem, fluorescent dye which a machine is using is always FITC. FITC stands for fluoroisothiocyanate. Fluoroisothiocyanate. So remember, in the patient you want to test for antigen, you add an antibody with the fluorochrome signal, reaction hoga, fluorescence aayega. So simple, if the antigen is present, the fluorescence is going to be present. Now this was direct fluorescent testing. What is indirect fluorescent testing? It is of two types. Indirect fluorescent testing mein you can test antibody or the antigen. You can test both and concept is same. If you want to test for antibody, what will you add from the patient? You will add antibody from the patient. If you want to test for antigen, you will add antigen from the patient. So the concept is the same. I just want to tell you that in direct fluorescent testing, you are directly measuring only antigen. Whereas in indirect fluorescent testing, you can measure both antigen and antibody. And I'll give you an example. FTA absorption. Right now, you might have studied bacteriology, so you would know. Fluorescent treponema antibody. It's a treponema pallidum test and naam se hai fluorescent treponema antibody. So that is an example of the Indirect immunofluorescence. Fluorescence word has come now. So direct for antigen, indirect for both antigen and antibody. Coming to the last test that we have in this chapter, and that is going to be the CART test. Of course, this is also an antigen antibody, and you can do CART test for so many things. I'm only giving you examples. You have CART test for hepatitis B, hepatitis C. There are CART tests for malaria. There, of course, how can I forget COVID? There are CART tests for even urine pregnancy test is a CART test. Why the blood? Urine pregnancy is also a CART test. So there are n number of CART tests. Typhoid matlab typhoid fever ka bhi CART test hota hai. So it's an endless list. What I want to tell you is, they will ask you what is the principle of this CART test and how do you read it. Principle is, it is based on ICT, immunochromatography. It is based on ICT, which is immunochromatography. You will have a control line always has to be there. Please remember, the control line always has to be present, because this tells you that everything is going normal in your lab. Sab kuch theek chal raha hai. This tells you the validity of the test. So this has to be there. But when the test line will be present, when the test line, or you can say when the test line is seen, it is a positive reaction. When the test line is not seen, it is going to be a negative reaction. So what are these lines seen on? Ma'am, ye andar dekho, peep inside. What is this white color strip? Yahan se this white color strip is starting. Because I know that I will be adding my blood over here. How is my blood trickling like this and going up till here? How capillary action se kaise glide kar raha hai? Because inside what you have is a nitrocellulose membrane. You have a nitrocellulose membrane. So repeating once again, what are the questions you get? CART test is done for a million things, you know that. Principle, question number 1, 2019 NEET PG question. Principle immunochromatography, that was 2019. After COVID, so this becomes a million times more important. Immunochromatography, what is the membrane made up of? It is made up of a nitrocellulose membrane. What does control line tell you? It tells you the validity of the test. And test line to pata hai, if it is seen, it is positive. If it is not seen, it is negative. We are going to start our journey with number 1, the complement system, the first K. Now, I could have called it complement pathway also, but it's not one pathway, it's a system. It's a system of 3 pathways. So please remember, there is something known as the very, very famous classical pathway. There is something called classical pathway. Then there's an alternative route. So there is something called alternate pathway. Right, there's some alternate route of starting it, alternate pathway. And then there is something called MBL pathway or lectin, or in other words, if you want the full form, mannose binding lectin pathway. Mannose binding lectin pathway. So simple, classical pathway, alternate pathway and MBL pathway. Let me tell you one thing, no matter whichever pathway you start, your end result is going to be a molecule called MAC. I'll tell you what is MAC. So first you'll say that, okay, if everything is the same in between them, then why are there 3 pathways? Because their starting point, what are they activated by, that is going to make a difference. What are they activated by? So number 1, the classical pathway is activated by immune complexes. What do you understand by immune complexes? Immune complex means antigen-antibody reaction. And which antibodies are going to come over here? IgM and IgG. Any immune complex that contains IgM and IgG will go and activate which pathway? The classical pathway. Talking about alternate, A for A, any immune complex containing IgA is going to go and activate the alternate pathway, A for A, simple. And when I talk about mannose, mannose is something in the bacteria. So any pathway which is directly activated by the microbial organisms, any pathway directly activated by the microbial organisms, that is going to be the mannose pathway. So simple, end result of everything is going to be the same. The starting is different. So classical by classical complexes, M and G complexes, alternate by A, IgA antibody complexes, and mannose by the bacteria or the microbial. Now let me show you what is the tough part going to be over here. So first and foremost, these are the three pathways which I've tried to put up over here. I'm going to tell you a shorter version of it, and then obviously I'll be telling you a detailed version of it. So let's start with the shorter version first. There is something known as the classical pathway. How do you activate the classical pathway? By immune complexes, by antigen antibody complexes. And can you see which are the set of molecules that you have activated first? The first molecule that I activated was C1. After that, I have a rule. First go for even numbers, then go for odd numbers. C1 will then activate all the even numbers, that is C4 and C2. They will get activated. After you have activated C4 and C2, what is the next number that you are going to activate? The odd numbers. So then you go for 3. After that, you go for activation of 5. That's a shorter way of learning it. So you start from C1, then you activate the even numbers, that is 2 and 4, and then you go into the odd numbers, that is 3 and 5, and half of your pathway is over. I want to give you obviously a complicated version of it. So let's start. When we start with the classical pathway, guys, obviously there was some antigen antibody complex that started it, and the first thing to get activated was C1, which formed C1q. Okay, QRS, many times it formed. I am interested in C1q. Okay, this C1 then went and activated C4 and C2. Now I just want to tell you one offhand thing. Whenever I'm saying something is getting activated, means it is forming an A version and a B version. Some of the things, some of the A's and B's I'll keep, some of the A's and B's I'll discard. Okay, but when I say something is getting activated, means it's being broken into A and B. So when I'm saying C4 is getting activated, means it's getting activated into A and B. Similarly, C2 is getting activated, it is getting activated into A and B. That's your basic concept. Okay, so remember, C4, I want to keep the 4B 2A. So how are they activated? They are activated into C4B2A. So now 4 activated, 2 activated, even numbers done. Move on to the odd numbers. Which is the one that you will activate? You will want to activate C3. So can I say who is activating C3? 4B and 2A is activating C3. Can I call it the C3 convertase? Can I call it C3 convertase? So C4, that is what was there in the previous one also, if you realized. They were together known as the C3 convertase because they were converting C3 into what? They were converting, again, when I say someone is getting activated, means there's going to be an A and there's going to be a B. This means C3 is now going to combine to this molecule. So what do you have? You have 4B, 2A, 3B. So BAB, alternate chalega. 4B, 2A, 3B is formed. 3 has been converted. 3 is a part of this complex now. After 3, what's the next odd number? The next odd number is C5. So can I say these 4, 2 and 3 that are converting C5 are now known as C5 convertase. And when I say it's getting activated, means C5A and C5B. I am now going to leave all the past 4B, 3, everything I'm going to leave. Now I'm only going to pick up the C5B. And you know what am I going to combine it with? I'm going to bring all its friends together. 6, 7, 8, 9. I'm going to pick up the C5B, combine it with the next 6, 7, 8, 9, and together they are referred to as the membrane attack complex. What is membrane attack complex? Membrane attack complex, whose membrane are you wanting to attack? The bacteria. I want to kill the bacteria. So is this going to go, and for example, if this is a dirty dark bacteria over here that I have made, is this going to go and break the bacteria? Yes, it is going to cause bacterial lysis. Whose membrane is it attacking? The bacteria's membrane it is going to go and attack. It's not a very easy complement system. You read it in weather path or micro, it's tough, but we'll try to simplify it. Let's repeat. The story starts off with C1. It has to start with number 1. First, I'm going to target all the even numbers. Then I'm gradually going to go and target all the odd numbers. So first I activate 4 and 2. So C4B2A is formed. That is now going to activate 3. So then what we have is 4B, 2A, 3B. That is now going to activate 5. And as soon as 5 is activated, 5B, 6, 7, 8, 9 are going to form the membrane attack complex and kill off the bacteria. Okay, having said this, coming back, you realize that your classical pathway is finally a tick mark. Let's talk about the alternate pathway. Alternate pathway is not going to start from that one and all those usual. Alternate means it's going to take some alternate route. It is going to start from C3. That's why it's alternate. It's taken some alternate route. So C3 along with a lot of factors, factor B, factor D, proper DIN, it is going to break. So please remember, I'll try to fit in the alternate pathway over here only. When I talk about the alternate pathway, the story starts with which antibody? With IgA, and it is going to start with C3. So please remember, C3 is going to be broken into 3A and 3B as always. Every molecule is A and B. So this 3B is then going to combine. This 3B is then going to combine with something known as factor B. Please listen to this very carefully. Factor B, factor D, and proper DIN. There is involvement of factor B, factor D, and proper DIN. And you know what happens? Especially factor B. Factor B also starts breaking like these numbers into BA and BB. So you know all the B's come together. I have something known as 3BBB. The 3B from here and BB from here, they come together and they make a complex called 3BBB. And you know whom are they going to go and break? Again the C3. They basically want to come and merge with this pathway at the C3 level. They again go and merge at the level of C3. So first C3 broke. Let's do a recap. First C3 broke into C3B. It took care of all the B's together. It's taken all the B's of its family 3BBB and has gone and attacked another C3. So now ultimately what is going to happen? If it attacks the C3 once again, is it going to result in the formation of 3BBB, 3B once again? If it's again going and attacking the C3, means it is once again going to form C3B, BB, 3B, and finally after that, that will go and attack your C5. And once C5 is attacked, it's going to be the formation of the membrane attack complex. You can have a look at it once again. Let's have a look at it again, guys. We started off our story with what? We started off our story with 3B. 3B acted upon with factor B, factor D, proper DIN, 3B, BB bana. That has gone and again acted upon on C3. So means C3B, BB, 3B. And once that complex is ready, that is going to go and act on C5. Once C5 is done, I have the membrane attack complex ready. That was the alternate pathway. Now I'm only left with the mannose binding lectin pathway, and that is neither going to start with C1 nor is going to start with C3. If I see, this is starting with the middle molecules and directly going at C3 convertase, means it's starting off with molecules called C2 and C4 and is directly going and targeting where? C3 convertase. So if I realize now which is the meeting point of all these pathways, be it classical or alternate or MBL, they are coming and meeting at the level of this C3 convertase, means they are coming and meeting at the level of something where I want to break C3. And that means what is the most critical step that I can talk about in the complement system? Please remember, the most critical step is a very favorite question, and that is the conversion of C3. The C3 conversion is the most critical step because all of them are going to come and sit down over there. Okay, having said that, I have a couple of questions for all of you, which is based on whatever we have studied up till now. Nothing new, but whatever we have studied up till now, let's see if you are able to answer it. Can you remind me, out of all of these, if I ask you, what is the composition of membrane attack complex? That is what they will be asking you. What is the composition of membrane attack complex? It is C5b to C9. So 5b, 6, 7, 8, 9. Question number 1, sorted. What is the composition? Or what is the next question? Which are all of these which are chemoattractants? If you remember, we had one question on chemoattractants which are going to cause chemoattraction, and one question was there on opsonins, right? So now we need to know those. Please remember, for chemoattraction, you had the A molecules, particularly C5A, which is a very famous question. Then, what else can the A molecules do? Any idea? What else can C5A, C3A, all these AA molecules, they are also going to be anaphylotoxins. They are also responsible for anaphylaxis. So remember, all the A molecules are anaphylaxis. All the A molecules are attractants, but C5A is the most potent attractant. And all the B molecules, C3B, 4B, 5B, all the B molecules were opsonins, right? Are we clear with this basic? Nothing new that I taught you, everything from what we studied. Now I'm going to teach you something new, because with this complement system, you also need to know all the diseases. So tell me, which is the most common complement deficiency known in humans? And that is C2. May or may not come across as a disease, but at least C2 is the most common complement deficiency. Okay, in someone, if the early complement proteins end up undergoing a deficiency, C1, C2, C4, that has been associated with an autoimmune disorder, that is SLE. That is associated with SLE. So early ones, SLE. If the late ones undergo a defect, that is C5b to C9, C5b to C9 was membrane attack complex, there is infection with many capsulated organisms, and one very famous infection is going to be Neisseria. Let me focus on this, very, very important, very famous infection that you need to know is Neisseria infection if there is MAC deficiency. Now comes the question that has come so many times in NEET, in FMG, in INICET. What is C1 inhibitor? So now, all said and done, everything was going, this pathway was going on and on and on, and in the end, membrane attack complex was forming everywhere, bacteria was being killed. Do you need something that should stop this pathway? Because if this keeps going on in my body 24/7, it will end up killing me only. It will end up killing my own cells. So when required, it should work, and when not required, it should not work. So they realized that you need some people to control this pathway. And that is when they came up with that if you do not let the first step occur, C1 is inhibited, then the entire pathway will not occur. So simple. If you want to control this pathway, do not let the first step occur. So for that, God has given us something called C1 inhibitor. C1 inhibitor controls the activation of C1. It controls the activation of C1. Basically, it controls the complement system. But now I'm talking about a disease when this controller is lost. If the controller is lost, then, like for example, if you are trying to get distracted, I am the one who's going to control you. But if I only go away, or if you switch off this app right now and send me off, then there's no control. Then you will be doing whatever you want. So if the controller is lost, big time problem. And that is going to be hereditary angioedema. That's the name of the disease that you all have to learn. It results in hereditary angioedema. And please guys, the way I've said edema, first thing you need to know, where is the edema going to present? So the edema is most commonly present around the eyes and the lips. So let's draw a funny looking diagram. So if this is a person, for example, and if they ask you where is the edema going to be, so the edema is going to be a weird swollen kind of an eye. Number one, the edema is periorbital, and second, the edema is going to be around the lips. So a very weird looking man that we had drawn, right? So remember, there is a periorbital and a mouth or around the lips. That is where the edema is there. All that is okay. When can this come as a medical emergency? Because that is a question that comes. What if the person ends up having in the neck, means larynx? If someone ends up having laryngeal edema, then this means he will not be able to breathe. If there is laryngeal edema, this means the patient has entered into respiratory distress, and that makes it a medical emergency. Run to the hospital. So remember, if the edema is around the eyes and lips, is still understandable, but if it occurs in the larynx or the respiratory tract, big time medical emergency. That is what has come in the question a lot of times. So remember, hereditary angioedema is because when the controller is lost, C1 inhibitor is deficient. When you're talking about this, why don't you talk about the others also? Let's talk about all of these. And you know what are all of these? All of these are controllers. That is what I want to tell you. All these things that are mentioned over here, CD55, CD59, factor H, factor I, CD46, all of these are controllers of the complement system. So, CD55 and CD59, what do they break? How do they control? They break the two most important odd number converters, C3 converters and C5 converters. So if these three are broken or these two are broken, complement system will not work. So they're good for us. CD55 and CD59 control your system. But just imagine if CD55 and CD59 go missing, the controllers are gone. Does it remind you of any disease in the blood? There is a disease called PNH, which means there is paroxysmal nocturnal hemoglobinuria. There is paroxysmal nocturnal hemoglobinuria that you have to know. Okay, next let's look at factor H, factor I and CD46. They are also controllers, but when factor H, factor I and CD46 go missing, it's a very important disease in medicine and pediatrics. That is going to result in atypical hemolytic uremic syndrome. Atypical hemolytic uremic syndrome. Finally, I want to correlate this with ophthal also. Have you guys studied that factor H is associated with something? Factor H defect in ophthal, what can you think of? Have you heard of ARMD? That is age-related macular degeneration. Age-related macular degeneration, that is because of factor H defect. So this is basically trying to tell you all the relations of complement systems that are going to come in the future in your life, be it in path or be it in other subjects. So remember, CD55 and 59 are PNH, factor H, factor I and CD46 are atypical HUS, factor H in ophthal is age-related macular degeneration. And for the time being, you just need to know that the same disease I will be telling you under, or the same set of things I will be telling you under kidney. Because now there is a disease called MPGN, which you have to know, which is very, very important. So please remember, for the time being, I can only tell you a little bit about it. MPGN type 2 is now said to be a complementopathy. It is said to be a complementopathy, means there is a problem in the complement system. What is MPGN? It's a disease in the kidney, membranoproliferative glomerulonephropathy. Membranoproliferative glomerulonephropathy is a defect in the kidney. So right now just know it's something to do with complement system, okay? Having said that, the complement system is done, and that finally marks the end of your entire immunology chapter. So like I said, everything